everyone, and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This series is brought to you today by Plantae, the open online community for plant scientists, powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who are not yet members of ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities that we provide at aspb.org. Many of you may have attended ASPB's annual plant biology meeting in the past. Each year, this event brings together plant biologists to talk about the latest research topics. In 2020, this event was held completely online and we had over 2,500 participants. ASPB's program committee is currently working on making plans for next year's event. The overall theme will be plants powering the bioeconomy. If you have ideas for workshops and would like to submit your ideas, please send us a proposal by December 4th. Although we are currently planning for a face-to-face meeting in Pittsburgh, organizers should describe how they would convert their workshop to a hybrid or a virtual conference format if that becomes necessary. I'd also like to encourage you all to apply for travel awards to attend the plant biology meeting in 2021. More information about funding opportunities can be found on our website. Notice that the first two deadlines are early in December. Today's talk is the 23rd talk in our Plantae Presents webinar series. We've had so many amazing speakers this year, so if you haven't already done so, do check out our archive and watch all of the previous talks in this series. You can find information about each event on our plantae.org blog and subscribe to our Plantae YouTube channel to get notifications when new recordings are posted. We have an upcoming panel all about specialized metabolites. This is a great chance for you to present your work in this series. All you have to do is submit a 7-minute video of your research. We will choose the top 4 videos to premiere live and then have speakers join in a panel discussion afterwards. These have been a really fun way to include more people and have more interactive webinars. If you work in this area, please send us a video. Also, if you have suggestions for new panel topics, let us know in the chat box. All right, so these are just a few of the opportunities that ASPB provides. Become a member, follow us on social media, and subscribe to our weekly newsletter to get more updates and opportunities directly in your inbox. If you do have to leave this webinar early, know that a recording will be made available within a few days. Today's talks will each be 20 minutes each. As you listen to each talk today, add your questions for the panelists in the Zoom question and answer Q&A section. Questions specifically for our panelists should be shared in the Q&A section to make them easier for our moderator to navigate. You'll also notice that there is a chat area open. This section is for you to introduce yourselves, say where you are from, and connect with your colleagues. All right, with all that information, now I'm going to introduce Ning Zheng. I think I got it. Ning Zheng is with us today to moderate today's session and share your questions with our speakers. Ning Zheng is a tenure-track assistant professor in the Department of Plant Biology at the University of California, Davis. Ning Zheng's research group integrates approaches from fields of structural biology, biochemistry, and plant biology to elucidate plant signaling pathways and reveal their regulation by the ubiquitin proteasome system. All right, thank you for joining us, Ning Zheng. I'm going to hand it over to you now to introduce our speakers and moderate today's session. Thank you.
All right, thank you so much, Katie. I'm going to introduce our first speaker. So it is a huge pleasure to introduce Dr. Ning Zheng, who's my mentor and friend, who is also an icon in structural biology, in particular in plant structure biology. So just a little bit about the background about Ning. Ning obtained his PhD from University of Texas. He then did his postdoctoral studies in New York at Sloan-Kettering Memorial Center. And already during his postdoctoral years, Ning made the groundbreaking discoveries by revealing the entire molecular architecture of one of the most intriguing ubiquitin ligases the SCF complex, which revealed how proteins are targeted for degradation at the atomic levels. And this is only one of the things that he did in his postdoc. And then he joined the faculty at the Department of Pharmacology at the University of Washington, where also I did my postdoc in his lab at Seattle, the beautiful Seattle, where he continues to make a cutting-edge research in the field of ubiquitin biology, as well as ion channels. And of course, in plant biology field, and just to name a few discoveries, as you all probably know, Ning and his group are behind the groundbreaking work of tier 1 oxi-AA and CoI-1 jasmonate complexes that not only solved long-standing questions in the mechanism of auxin and jasmonate signaling pathways, but also serves as inspiration to increasing number of biopharma companies that utilize this Tegon paradigm that coming actually from plants, which is pretty remarkable. So it is not surprising that Ning received several awards during his outstanding career, including the Pew Serial American Association for the Advancement of Science, and he has been an investigator of the Howard Hughes Medical Institute since 2008. So today, Ning will share with us his latest exciting research on the structural basis of salicylic acid signaling by NPR proteins. And without further ado, let's welcome Ning Zheng to the stage.
Thank you, Ning Zheng, for the very generous introduction. I will share my screen. Can you see my screen?
We sure
Yes.
can. Great.
All right. Okay, I would like to first thank Javier and the rest of the steering committee for offering me this opportunity to present our latest study on the structure basis of salicylic acid perception in plants. Before I start, I need to make a confession. That is, I'm not a plant biologist by training, but instead, I'm more of a plant biologist wannabe. Our lab actually belongs to a pharmacology department within School of Medicine at the University of Washington, focusing on structural biology of protein-protein and protein-small molecule interactions. Let me turn on my laser pointer so that you can see my cursor clearly. Our lab actually became interested in plant hormone more than 10 years ago because of our longstanding interest in ubiquitin proteasome system. And I hope to this audience, I don't have to explain ubiquitin ligases in great details. They are the enzymes acting at the last step of a 3-enzyme cascade, E1, E2, and E3 enzymes, basically dictating the specificity of protein ubiquitination and degradation. So timing obviously is very important for protein ubiquitination and degradation, and we are particularly interested in how cellular signals control the interaction between the substrate and the E3 ligase. As you know, that among a handful of known plant hormones, which regulate almost every aspect of plant physiology, about half of them actually doing so by acting as the signal controlling E3 ligases. And this paradigm was first established by the groundbreaking discoveries of auxin receptor FOX protein tier 1 made by Dr. Mark Estelle, Alvin Leiser, and Stefan Kapinski back in 2005. So since then, we have seen variations of the same theme in other plant hormones, such as gibberellins, jasmonate, and lately stragolactones, and each with its own unique mode of action. So salicylic acid represents yet another phytohormone which might act through ubiquitin ligases. Although I have to say that there seems to be multiple layers of mystery that we have to unveil in order to truly understand its mechanism of action. So our work on salicylic acid, or short for SA, started in 2011 when I met Dr. Xinlian Dong in Duke University. So in the past 2 years, it has been a very productive and lovely collaboration between the two groups. On the Duke side, John Withers, and together with other lab members in Xinlian's group, played a key role in characterizing SA and SA receptor system in vitro and in Plantae. Well, our group actually struggled for nearly a decade trying to make progress on the structural biology end. Here, before I venture into the story, I would like to acknowledge numerous lab members, particularly Dr. Wei Wang, Dr. Heng Li, and previous members, Jim Rubble and Wei Manxing, who actually really exhausted many approaches before we finally got the first glimpse of the hormone in action. So many of you know salicylic acid is a plant immunity hormone, which is generated upon local infection by pathogens such as fungi, bacteria, and viruses, and it's transmitted systemically to distal sites to establish immunity. This is a process called systemic acquired resistance, which can persist for weeks to months after the initial primary infection. So the role of SA in this process was illustrated in early 90s and can be appreciated through this figure on the right, which depicts the rise of SA level in uninfected leaves after 2 days when the primary site was infected by tobacco mosaic viruses. So in these uninfected tobacco leaves, SA subsequently induced the expression of a number of pathogenesis-related genes, which contribute to disease resistance. And here, the expression of PR1 gene is visualized by northern blot for PR1 messenger RNA 5 days after the primary infection. So in 1994, Xinlian's group actually isolated a Arabidopsis mutant called NPR1, non-expressor of NPR genes 1, which carries a single recessive mutation that abolishes SA-induced expression of PR genes. This finding revealed a positive regulatory role of NPR1 in SA signaling. Now, interestingly, in Arabidopsis thaliana, there are multiple PR1 paralogs which share the same domain composition. Subsequent studies from Yuelin Zhang's group in UBC in Canada has shown that NPR3 and 4 seems to play a negative role in regulating SA's pathway. Here in the NPR3-4 double mutant plant, the basal level of PR1 gene is elevated. So now you have actually multiple NPR proteins regulating defense-related gene expression in opposite ways. How they do this? Well, at the sequence level, all NPR proteins contain the N-terminal BTB domain, and this domain is frequently found in the family of Ubiquitin Ligases. So in light of several other plant hormones being perceived by E3 ligases, it is perhaps not a far-fetched idea that NPR proteins might function as the missing SA receptor. Indeed, this turned out to be true, although the receptor system seems to be far more complex or convoluted for SA than other plant hormones. In 2012, Xinlian's group actually reported robust SA binding activity for NPR4, but not for NPR1. They also found that NPR4 and NPR1 can form a complex which can be disrupted by SA. So a model, therefore, was proposed to reconcile the opposite role of NPR1 and NPR4 in SA signaling, in which NPR4 acts as a SA-sensitive E3 ligase to degrade NPR1. Intriguingly, using a different SA binding method, the press group in Brock University in Canada was able to detect SA binding by NPR1, therefore designating it as the SA receptor. Yuelin Zhang's group in UBC recently further proposed a model in which NPR3 and 4 acts as transcription co-repressors, whereas NPR1 functions as an activator. Their activities are either inhibited or activated by salicylic acid. So it is most likely that these two models reflect different aspects of the system, and it is our belief that structure studies, and particularly structure-guided mutagenesis analysis, will be very helpful, if not critical, for fully dissecting the mechanism of action of SA and the functional roles of individual NPR proteins. So From the get-go, actually, we were able to purify full-length NPR proteins from both bacteria and insect cell, and we obtained beautiful crystals, but only ended up being very disappointed by their poor X-ray diffraction, which prevented us from making progress for several years. Now, it turned out that this problem was encountered not only by us, but by other groups. This forced us to go back to the drawing board and take the conventional divide and conquer approach by first mapping the SA binding domain of NPR proteins. So the most classic method for mapping functional domains is by limited proteolytic digestion in structural biology. So we soon realized that NPR4 protein displayed SA-sensitive proteolytic digestion pattern. As you can see here, in the presence of SA, not only NPR4 protein is more stable or stabilized, but also upon trypsin digestion, this protein yield different digestion pattern. So taking advantage of this phenomena, we made several constructs of NPR4 and was able to derive the minimum fragment of this protein that retains SA sensitivity. And we call this fragment SBC, stands for SA binding core. So this, of course, is a classic approach, but we are in 21st century, and we're actually equipped with far more sophisticated technologies, one of which is hydrogen deuterium exchange mass spectrometry. So in this method called HDX-MS, you basically transfer your protein from H2O to a D2O solution, and then you quench and digest your protein at different time points and subject them to mass spectrometry analysis. So solvent exposed labile hydrogen atoms on your protein will be displaced by deuterium, and their different structural contacts will determine their exchange rate. So if your protein binds to a ligand, the ligand might actually physically protect certain hydrogens from being exchanged, or the ligand might induce conformational changes to alter the exchange rate of these labile, these otherwise labile hydrogen. So we took the full-length NPR4 protein and did HDX analysis in both the presence and absence of SA. So I don't have time to explain the details of this rather complex plot, but I just want to draw your attention to the lower part of this graph, which is derived by subtracting the SA-free exchange profile from the SA-treated sample. So this diagram actually reveals the region of the polypeptide highlighted in yellow, whose exchange rates are altered by SA. I hope you can tell that consistent with our proteolytic mapping experiment, this HDX-MS experiment also told us that the SBC region within the C-terminal NPR1-like domain of NPR4 is most likely responsible for binding SA. So equipped with this information, we actually went on, finally managed to purify and crystallize this NPR4 SBC fragment after refolding it with SA present and determined its crystal structure at high concentration. So in front of you are the two orthogonal views of SA-bound NPR4 ligand binding core. And I hope you can appreciate that the SA binding site is constructed by 4 alpha helices, which resemble 2 interdigitated nutcrackers. So together, these 2 nutcrackers actually sequestered hormone right in the middle of the SBC domain. When I say right in the middle, it really is at the center of the domain. When we take a closer look at the SA binding site, we realize that SA is completely surrounded by NPR4 residues. One of them is the arginine 419, which actually neutralized the carboxy group of this 2-hydroxy benzoic acid compound. So if we actually put NPR4 protein in surface representation and peel off the outer shell, you will realize that the receptor actually literally creates an internal cavity that encapsulates the hormone within. And we did not see, surprisingly, any ligand entry or exit path. In other words, the hormone is completely buried in the center of this protein. So we know this is not an artifact of the fragment we study or the refolding process or the crystallization condition, because the SA binding region revealed by the crystal structure actually matches exactly the same residues that are most sensitive to SA as revealed by our previous hydrogen deuterium exchange experiments using the full-length NPR4 protein. Okay, so here what's colored in dark are the residues or regions of the protein polypeptide in the entire full-length protein that's most sensitive to SA. So what does this rather unusual, unexpected structure features mean? Well, this result strongly suggests that NPR4 SA binding domain must undergo major conformational changes upon binding to the hormone. In other words, without the hormone, the 4 helices must adopt an open conformation that expose the SA binding site, make it accessible to the ligand. Upon binding to the hormone, binding to the NPR4 hormone binding domain, SA literally nucleate a hydrophobic core and close the domain structure. In addition to this conclusion, we actually also noticed that the majority of SA contacting residue in NPR4, which are highlighted in yellow, are actually conserved in NPR1 orthologs, okay, based on sequence alignment. So this suggests that NPR1 at least is equipped with the proper amino acid for recognizing SA. And this prompts us to revisit the SA binding experiment using recombinant NPR1, NPR4 proteins. So after consulting with Yuelin Zhang in UBC and adopting their SA binding method, we indeed were able to detect SA binding activity using full-length NPR1 protein. However, when we estimated the receptor occupancy for NPR4 and NPR1, we actually saw a dramatic difference between the two proteins. In this particular assay, we estimated that 8% of NPR4, recombinant purified protein, was occupied by SA when we started with saturating amount of ligand. By contrast, only 0.02% of NPR1 is in the SA bound form, despite the fact that we supplied plenty hormone. Okay, so the next immediate question we ask is whether NPR1 really used the SBC region to bind the ligand. So it turns out that's true because we were able to purify NPR1 SBC fragment in complex with a NPR1 binding protein called Nimin2. And we certainly can detect specific binding that's dependent on the equivalent arginine in NPR1 for binding SA. And the binding affinity actually matches the full-length protein. So, but similar to the full-length protein, the NPR1 bound fraction, the SA bound fraction of NPR1 is also extremely low in the 0.02 or 0.03 range. Okay, so what does this tell us? Well, we think that based on these results, we can draw two conclusions. First, we think we hypothesize that the apo form of NPR proteins most likely are structurally dynamic and are in equilibrium between multiple structural states. We think that at least one state in which the ligand binding pocket is inaccessible to SA, that explains why a major fraction of the protein are not SA bound or not capable of binding SA in our assay. Second, even though NPR1 share almost identical amino acids with NPR4 in the SA binding pocket, these two proteins seems to be categorically distinct in the sense that the predominant species of NPR1 in the recombinant protein is not ready to recognize this ligand. Okay, so now we actually revealed a potential conformational change of NPR4 SBC. What is the biochemical consequence of these changes? After all, these are just in vitro structural studies. Now remember, in Xinyan's group, actually, they proposed this model where NPR4 interact with NPR1 and ubiquitinate it as a substrate. So we actually first started by validating this NPR1, NPR4 interaction using a GST cooldown assay. Okay, So in this GST pull-down assay, followed by Kumasi staining, we indeed detect robust interaction between NPR1 and NPR4. As a structural biologist, we of course wanted to be more quantitative. So we developed a so-called alpha screen assay to measure the affinity between the two proteins. So alpha stands for amplified luminescent proximity homogeneous assay, quite a mouthful word. But you basically have two affinity tagged binding partner proteins immobilized on either donor beads or acceptor beads. And the two beads will come in close proximity due to protein-protein interaction, and you will see luminescent signal. So using this assay and tag-free NPR1 as the competitor, we can estimate the IC50 or close to KD of the interaction pair. And that turned out to be in the single-digit nanomolar range. So this binding is very, very tight. Now, in addition to the direct binding, we also confirmed that in the presence of 100 micromolar SA, the interaction is compromised or significantly impaired. And to be more quantitative, we could take the same alpha screen assay and obtain a dose response curve of SA in disrupting the NPR1, NPR4 complex. And that actually revealed a rather impressive nanomolar potency of the hormone. And to show that this effect is specific, we replaced SA with the inactive SA analog benzoic acid, which lacks the hydroxy group at position 2. And this compound actually only showed marginal effect at a much, much higher concentration. Okay, to further validate the specificity of the effect, we actually included both wild type and 2 single amino acid mutants of NPR4 in the GST pull-down assay. Remember, this arginine 419 is critical for neutralizing the carboxy group of SA. Its mutation to glutamine completely abolished SA binding, and accordingly, when we use this mutant in the pull-down assay, the interaction is insensitive to SA. By contrast, another single amino acid mutation at threonine 488, which actually did not impact SA binding activity in the SA binding assay, retained the sensitivity of SA's toward SA. So interesting enough that I did not include the data on this slide, but the disruptive effect of SA is dependent on this single arginine amino acid on NPR4, but not on NPR1. In other words, if we replace NPR1 with NPR1 arginine mutant that cannot bind SA, it does not change the dose response curve of SA in disrupting the two complexes. So I hope that from this experiment, I convince you that the two proteins, NPR1 and NPR4, do interact with each other. And the conformational changes of SA induced by SA binding actually will block NPR1 interaction, basically responsible for dislodging NPR1 from NPR4. So now at this point, let's not forget that these cartoon drawings are missing 2/3 of the proteins. After all, our structure only focuses on a fragment of NPR4, and we're missing a large part at the N-terminus containing the BDB domain and ankyrin repeats domain. From our hydrogen deuterium exchange experiment, we know that SA sensitivity actually is detected in the small region in ankyrin repeats. And this effect is very repeatable and can be detected in 2 independent peptides. So this suggests that the 2 domains or the 2 halves of the protein most likely will communicate with each other. Okay, to further probe this idea, we actually made a series of chimera proteins between NPR1 and NPR4. And remember, NPR1 has very weak SA binding activity. NPR4 has much higher activity. And by swapping the domains, we can demonstrate that the N-terminal domains, including the BTB domain and the ankyrin repeats, can actually impact the SBC's region's ability to bind SA. And I won't actually go into the details to explain this figure. And if this is true, then we think that effect must be communicating through the surface residue of SBC, not the residues contacting the SA. So here we actually chose several surface residues from our structure and plug them in, purify the protein, plug them then into our assay binding assay. And we noticed that a single amino acid mutation of this surface phenylalanine residue actually severely impaired assay binding activity in NPR4. By contrast, a threonine amino acid that's located in the invisible loop or flexible loop in this domain structure enhanced the assay binding activity. Now, most strikingly, when we combine these two mutants, we actually have a far hypersensitive mutant that has much stronger assay binding activity. So these unexpected findings really indicate that we can perhaps fine-tune assay sensitivity of NPR4 with subtle amino acid changes. Now, to test whether we can translate these in vitro binding results into in vivo functions, we actually first monitored the stability of endogenous NPR1 in the cell-free protein degradation assay using different transgenic lines containing different versions of NPR4, either the wild type, the hyposensitive mutant, or the hypersensitive mutant. Keep in mind that NPR4 is proposed to bind to NPR1 and promotes its ubiquitination degradation. Assay disrupt this interaction and would stabilize NPR1. So as you can see from the figure on the left, without NPR4, NPR3, the endogenous NPR1 is rather stable. And with wild type NPR4, it is slightly destabilized. With the pretreatment of SA, you can see that the hormone hypersensitive mutant of NPR4 can barely touch NPR1, whereas SA-Hypo mutant can degrade NPR1 much faster in comparison to wild type because it's resistant to the action of SA that disrupt the complex. So now based on this degradation model, stabilization of NPR1 by SA should lead to upregulation of defense genes, right? The PR gene expression. So using the transgenic lines, so we can actually quantitatively measure the expression level of PR1 gene, and we saw a perfect correlation between PR1 gene expression and NPR1 stability shown on the left. And more importantly is that when Zhang in Xinlian's group actually challenged these transgenic plants with bacterial pathogen, he saw the protective effect of SA treatment was lost in the hyposensitive mutant in comparison to the wild type NPR4 protein, but it's actually the effect is markedly enhanced in the double mutant, which is hypersensitive to SA. So these results are quantified in the figure shown on the right with the dark columns. So overall speaking, these exciting data really support the inter-domain communication within NPR4 through SBC surface residue, but they also really highlight the promising potential of using structural information to engineer the SA receptor for enhanced plant immunity. Okay, to summarize, so we mapped the SBC as a SA binding core. We solved the crystal structure, and the SA is encapsulated in the middle of this binding module, hormone binding module, which implies that major conformational changes must happen due to SA binding. We confirmed NPR1, NPR4 interaction that's sensitive to SA, and we think that conformational change probably explains how SA can disrupt this interaction. We show that NPR1 has a much, much lower SA binding activity than NPR4, and we actually really need to reconstitute this ubiquitination assay to understand why NPR1 is the substrate, but not NPR4. Also, that we show that the BDB anchoring repeats can affect the SBC activity, and assay sensitivity of NPRs can be engineered and fine-tuned. Obviously, we haven't really visited this second model raised by Yuelin Zhang, where the NPR3 and 4 works as co-repressor and NPR1 works as a transcription activator. We need to understand how NPR1 and NPR4 interact with the transcription factors of TGA and how their activity is affected by SA. And that's going to be the next lines of research. Our preliminary data, however, show that NPR1 can indeed strongly interact with TGA, but that interaction is insensitive to SA. So we think something is missing in this diagram. With that, I conclude my talk and welcome any comments and questions.
All right. Thank you so much, Ning. That was fantastic work. Let me see. Cannot. Oh, I can have my video. All right. So I'm waiting to hear. So I have a few questions. So one question is from anonymous attendee. So does it mean that NPR 3 and 4 is high when there is no infection by pathogen?
The NPR 3 and 4 proteins are relatively stable. So it's the NPR 1 protein whose level actually would change due to, due to the degradation by NPR 3 and 4.
All right. So I will wait for more questions, but I have a question. So it seems really complicated when you think about the ubiquitination pattern. So I guess the vision is that NPR4 is undergo this conformational change that might allow it to bind the NPR1 at some sort of low concentration of salicylic acid. But once you have increasing concentration of salicylic acid, then it might also trigger the conformational change of NPR1, and this complex might fall apart. Is that a good assumption? And if it is a good assumption, can that maybe explain something that relate to maybe ubiquitination site that could be revealed?
Yeah, that's a great question. Obviously, number one, we don't really have the structure of the full-length proteins, so we can only hypothesize. So we think that the predominant sensor within the NPR1, NPR4 complex is NPR4 itself. Because number one, the hormone binding site in NPR1 is not responsible for the disruptive effect of SA. Okay, so it's only the NPR4 arginine residue can actually abolish that effect. In terms of conformational changes, we know that the SBC fragment must undergo conformational changes, but we don't know the grand scale conformational changes within NPR4, obviously. And what made, let's say, NPR1 serve as the substrate, but not NPR4, because both proteins has BDB domain. Both proteins could be the adapter of the choline E3, choline 3 ubiquitin ligases. That's a completely open question that we cannot answer at this point.
Right. Thank you. Okay, I have two more questions. One from Facundo Romani. He asked, are NPR1 and NPR3 and 4 conserved as antagonistic pairs in land plants, including the relevant residues that you identified?
That's a fantastic question. This is a burning question we need to answer. So according to Yuelin's model, he actually proposed that NPR1 lacks a eared motif that's found in NPR4, and we're talking about the Rhabdopsis thaliana paradox. And that ear motif is, even though it's missing in NPR1, in the Rhabdopsis thaliana, it's actually present in other species. So obviously, ear motif most likely is responsible for recruiting repressor complexes. But because of this lack of, let's say, this unique feature of Rhabdopsis NPR1 lacking this ear motif, it almost suggests that the function of NPR1 in Rhabdopsis is rather idiosyncratic, almost suggests that. So we really need to test all these models in other species as well. That's a great question.
Okay, I have actually a few more questions, but I don't know if we have enough time. Katie, can we do more questions or we should move on?
Yeah, I think maybe one more, and I can
Okay.
send the leftovers to.
Okay, so I have several more questions. One question from Shunyan Xiao. Elegant talk, he said. I have a question that may be more appropriate for Xinyan to address. Okay, so any natural mutations in these key residues that impact the SBC-SA binding in the Arabidopsis genomes, pangenome, to help individuals accessions to better adapt to specific path or environmental context through NPR1, NPR4 SA interaction?
Yeah, that probably is a question,
Yeah.
you know, for Xinyan to answer.
Okay, so maybe last one question that is short. Does SA affect interaction of NPR4 to choline?
Great question, actually. So we have been trying to reconstitute NPR4 choline interaction, and so far we have not been successful in terms of accomplishing this goal. So the interaction, the plant choline protein is very difficult to purify for some reason, and we could not actually test the effect of SA on NPR4 choline 3 interaction at this moment, but that's definitely something that we need to do.
Interesting. All right, so thank you so much, Ning. I will applause. I guess everyone do it at home, at their home. And now I'm gonna introduce our next speaker. So it is my great pleasure to introduce Dr. Destiny Davis. Destiny obtained her PhD here at UC Davis with the mentorship of Dr. Georgia Drakakaki. I actually have met Destiny some time ago as part of collaborative work with Georgia. And indeed, Destiny is a fantastic plant cell biologist and an expert in plant cell walls. She's currently working as a postdoc with Dr. Jenny Mortimer at JBay Berkeley Labs. And today, Destiny will present her latest research on callose, cytokinesis, and karyophyte. And without further ado, let's welcome Destiny to the stage.
All right, can everyone see my screen?
Yes.
Okay. All right, hello everybody. My name is Destiny. Today I'm going to be telling you a bit about what I did at kind of the last end of my PhD work, working with Georgia. Also, this is my current email address, and also you can find me on Twitter. So I did my PhD at UC Davis with Georgia Drakakaki, where we are largely interested in the intersection between endomembrane trafficking and the cell wall. So they're kind of like two different halves of that. So how endomembrane trafficking plays a role in cell elongation and wall maintenance during that process. But I was more focused on endomembrane trafficking, specifically focused around cytokinesis. So this here is a 4D video, and you can see a couple different instances of cell division happening. The green is a cell plate, an intermediate cell wall structure marker. So you'll see some of them initiate and then they'll be turned over as more and more vesicles arrive to the center. And then you can start to see them with the FM464 dye, which labels plasma membrane. Just to recap. So for cytokinesis, there still are many questions that are left unknown, specifically around the molecular details of these different events that we're seeing. Obviously, it's a very dynamic, very spatially and temporally regulated process, in that you need to flush all of these things, all of the cell wall material to the plane of division at a very specific time in order to have successful cytokinesis. So, as you can imagine, whenever you take a traditional genetic approach to kind of dissect this process and the different proteins that are involved in cytokinesis, you run into, frequently run into issues of lethality. In that if you start messing up genes that are involved in an essential process, just kind of, you basically just get no plant to study. So our approach to get around this and to circumvent issues of lethality and also redundancy is to use chemical inhibitors. So basically just to summarize, you have a gene, normal protein, get a phenotype. Classical genetics, you mutate that gene, get an aberrant protein, get an abnormal phenotype, which you then obviously study. In our approach, we actually use a chemical that we incubate with the protein, and that will lead to an abnormal phenotype. And this has been very useful in a bunch of studies, but specifically for us, it is conditional in that you can add the chemical at whatever developmental time frame that you want to get your phenotype. And it's tunable in that you can change the concentration and increase or decrease the severity of the phenotype. And in some cases, it's also reversible. So you can also get more information about like washout experiments and see how the plant recovers. So just to recap on plant cell division, it starts with Golgi-derived vesicles coalescing at the plano division, where they immediately begin to fuse and tubulate. And as you get further and further along in cytokinesis, these tubules start to puff up to create the cell plate lumen. The cell plate is this intermediate structure as more and more vesicles are arriving at the leading edge. And then finally, at the end, you end up with a fenestrated sheet of membrane that's traversing the plane of division that eventually will mature into a primary cell wall. The event that I focused on in my PhD was this essential deposition of callose. It's a beta-1, beta-1,3 glucan that's deposited at this transition between the setup of the membrane and then also the maturing into the cell wall. And so it's deposited in the lumen of the cell plate, and then it kind of disappears. It presumably is degraded as the cell plate matures into a cell wall. So it's very transient. However, whenever you get rid of callose, and we do this, our chemical is endocytin 7 or ES7. And so whenever you incubate dividing cells, or in our case, Arabidopsis root tips with ES7, it specifically gets rid of the callose at the cell plate. And what you're left with are cell plate stubs. And so you get cell plate breakages that will eventually persist, and you just get a bunch of cytokinesis defects. So this is kind of more the first half of my PhD, looking at the plant side of things, trying to elucidate the different molecular players that are involved in the callose deposition aspect of cytokinesis. About halfway through my PhD, however, I got interested in the evolution of de novo cell wall formation after reading a bunch of papers looking at algae cytokinesis and the myriad different patterns and processes that are kind of either in between, like phragmoplast or a phycoplast, whether or not they have a cleavage furrow or have a formal cell plate, and wondering how callose might fit into that story, the migration of plants to land. So when I started to map a couple of cytokinesis traits onto a phylogenetic tree, it was really interesting to me because callose present at the cytokinesis junction is kind of an early trait that evolved, but it had only ever been found in multicellular species, which was really curious to me because if you look at the Zygmatales group, it involves a lot of species that have a lot of examples of reduction. So they don't have flagella, they don't have plasmodesmata. A lot of them are unicellular, either unicellular or filamentous. And so if they're really that closely related to land plants, then why would their cytokinesis strategy be that much different, given that callose is so essential for cytokinesis in land plants? So in order to get at that question, I looked at Penium margaritaceum, which is a unicellular freshwater desmid. Here, it's very simple morphology. It just has two kind of semi-cells with the nucleus in the center. This center region is a very interesting region of the membrane and also the cell wall. It's called the isthmus zone. And here is a schematic kind of blowing up of the isthmus zone. And so basically the cell wall is very, very similar to land plants in polysaccharide composition. So what you have is an inner layer of cellulose, and embedded within that, there is this homogalacturonin lattice that is extruded through the cellulose layer to form these beautiful peaks, these beautiful like mountainous lattice along the outside of the cell. All except excluding this isthmus zone region, which I'll revisit later on. So using Penium, it's kind of emerging as a new cell wall model. The genome was actually just recently sequenced by the Rose Lab and with help with David Domazich. So with all of those tools in hand, I decided to, well, what if I treat Penium with ES7, what might happen? So here's a quick video of it dividing. Oh, is it gonna play? I might need to use my pointer. I don't think. All right, well, it was a video of it dividing. I'm gonna continue though. Okay, so long story short, ES7 causes growth defects in Penium cultures. So if you look at, this is a graph of Penium growth over 7 days with different concentrations of ES7. The highest concentrations being 7.5 micromolar and 10, which are the green and red bars here, or lines here. So as you can see, it has a significant effect on the overall growth of the culture in a concentration dependent manner, which is a similar phenotype to as we see in growing Arabidopsis roots. It's very concentration dependent. So this was my initial experiment. And so upon seeing this, I got kind of excited. So whenever I looked at the cells in these cultures, I did in fact see evidence of cytokinesis defects. And so Cyto9 is our nuclear dye here. And so normally it's not dividing one nucleus, but there are these two phenotypes that I saw in a bunch of about 90% of the cells that I treated with ES7. One of them is just an elongated isthmus zone where it literally looks like the isthmus zone had just been stretched out, and it coincides always with two nuclei. And then another one is fused daughter cells. And this one I characterize as fused because you can see some septum formation. They are just stuck together cytoplasmically, again with the two nuclei. So I took this to be evidence of cytokinesis defects, which we know from our plant work that callose is essential for cytokinesis in land plants. And I know from the literature, at least what I was reading, is that callose has never been shown to be important in cytokinesis or present in cytokinesis in unicellular plant species. However, here I see a cytokinesis defect when I use ES7, which is very specific for cytokinetic callose. So the obvious next step from this is to just look at callose. Maybe it was missed. Long story short, callose is absolutely there in Penium. And so on the left here, this is using an aniline blue dye. And so you see these are kind of the ends of the tips of dividing cells that have yet to physically separate. And so you can see there's beautiful callose caps on either tip of the daughter cell there. And what I didn't expect was to see such a dynamic progression of callose patterns throughout cytokinesis. And so starting here over on the left, the top is a 3D view. Some of them are tilted, some of them are not. And then the bottom row is a surface rendering of the 3D images. So here you can start to see a callose ring as early as this stage before you even get like daughter nuclei division and migration to their new isthmus zones. You see a single ring of callose all the way through until you have the daughter nuclei migrating to their respective isthmus zones. And then something really interesting happens in which this single ring kind of transforms into two rings. Then you see this puncta or foci appear in the very center of those two rings, kind of like a bullseye. And this will be the last physical place where the two daughter cells are touching. And then as you progress further, the rings seem to disintegrate or degrade a little bit as the center spot or foci region elaborates to form these beautiful callose caps that will continue to grow until the daughter cells physically separate, in which the callose sticks around for a little bit, but then it eventually goes away. And this, again, was all using the aniline blue dye. We also have confirmed this using the anti-callose antibody, which showed the exact same patterns that we saw in the aniline. So whenever I treated with ES7 and looked for callose, lo and behold, I was unable to detect it using either the dye, aniline blue, or the antibody, which was very exciting. That is, we found callose in this unicellular species, and also ES7 seems to have at least, a somewhat similar mechanism in Penium as it does to Arabidopsis. So the next thing I was interested in exploring is, what does this do to the cell wall? We know that the isthmus zone, being a very important region for not only cytokinesis, but also cell wall secretion and excretion, all of the cell wall material is secreted at the isthmus zone and then displaces the older wall towards the tips. So there's a lot of really interesting endomembrane trafficking and cell wall maturation happening at the isthmus zone. So I wanted to know if ES7 had an impact on the cell wall and what that might look like. So I looked at pectin. This is using the GIM5 antibody, which primarily labels the more mature pectin around the entire cell, except for this region at the isthmus zone. So the idea is that as pectin is excreted in the isthmus zone, it then gets unesterified, and then it forms this nice pectin lattice. So in the normal condition, you see it covers, the antibody labels the entire cell, and as you go through cytokinesis, you see this gap form. However, under ES7, it looks normal under non-dividing conditions, but when the cell begins to divide, you see labeling in the isthmus zone, which is completely opposite of what you see in the normal condition. And you also see an irregular kind of lattice pattern and sometimes some like clumps, it appears. When you switch to looking at GIM7, which primarily labels the pectin that is first secreted, so the deposition pattern is just kind of flipped for this one, and that you see GIM7 labeling primarily in the isthmus zone under normal conditions where it's not dividing, and as it elongates, you see it actually expanding to form this wide isthmus zone band. And then even further during cytokinesis, when the cells, you start to see the membrane seam between the two daughter cells, you see that band transform into two small narrow bands, one for each daughter cell. And that labeling on the tip of each daughter cell will persist for a little bit after the cells has physically separated. However, whenever you introduce ES7 into the system, you get normal labeling in interphase. It does not affect deposition during non-dividing cells. However, whenever you start to see the cytokinesis defects from the ES7, you get no more GIM7 labeling in the isthmus zone. And we are interpreting this, or we interpreted this to be kind of a compensation, potentially a compensation mechanism, in which maybe pectin that is deposited in the isthmus zone in a dividing cell treated with ES7 will be more quickly deesterified in order to kind of stabilize that isthmus zone area as it's trying to divide. Also, we can't rule out that ES7 could be doing something, the absence of callose could be changing the underlying isthmus zone structure in a way that disrupts cell division and the wall. So thanks to the recently sequenced genome, we were able to pull out 15 different Penium protein sequences that aligned to callose synthase 2 in Arabidopsis. And whenever you align them with the other, with callose synthases across different species, across the plant kingdom, you start to see that these four here are the most similar, and they have a similar domain structure. And so they do seem to have some conserved domains, specifically the glucan synthase domain, which is the biosynthetic domain for callose, with the other callose synthases across plant species, which just confirms our labeling experiments to show that, you know, what we have here is in fact callose. I will say it will take further studies to identify which one of these might be responsible for the callose that we see during cytokinesis, but it does confirm to us that it is there. So with all this information, we were able to put together an updated model of Penium cytokinesis that included an essential role for callose during cell division. And so one that starts with a single ring at these early stages, even prior to nuclear division, and that goes through a significant transformation in morphology change as the cells go through cytokinesis. And what each of these patterns is doing is yet to be explored, but it certainly leaves a lot of questions to be answered. And I'm also kind of putting forth a hypothesis that the callose foci that I'm seeing in between the two rings here, I think might be suggestive of a cell plate in that region. And the only reason I say that is because with my studies on Spirogyra, which is very closely related to Penium, it does seem to have, it has both a septum and a cell plate. And so I think Penium could be another example of that kind of mixture between cleavage, but also cell plate formation. And then whenever you add ES7 into the mix, quite simply, you get no callose and you get fused daughter cells, which then kind of feeds back into the cell wall, where you get some of that more mature pectin lattice deposition in the isthmus zone where you normally wouldn't see it. And just very briefly, Nissan mentioned that I am now working at JBay, and so I just wanted to give a little teaser for my work with Dr. Jenny Mortimer. So what we are doing is we are exploring rhizobia colonization patterns on plant roots. And so we're using these fabricated structures called ecofabs in order to grow Arabidopsis into these sterile environments, where I will then inoculate with a rhizobia community and then quite simply image them. So these are a collection of fluorescent labeled bugs that I then use a confocal in order to identify different patterns of colonization. And you know, I just started in the lab a couple months ago, and so I just now got my feet underneath me. And I'm really excited to continue this project. There's so many different areas in which we could explore, including cell wall composition, root hair density. We could use a myriad of mutant populations in order to explore what are the most important factors in determining the pattern of colonization. But I'm just not getting started, so just stay tuned for, hopefully I'll have something really interesting to say this time next year. With that, I want to thank you all for listening. I want to thank Georgia, obviously, my PhD advisor, all of my lab members, my dissertation committee, of course, and then specifically all of our wonderful collaborators without whom I could not have gotten nearly this far in my Penium work. And then, of course, I know I've been blessed to work with a lot of really fantastic people in the lab, so I just want to thank them too. And I would be happy to answer any questions.
All right. Thank you so much, Destiny. It was a beautiful talk. I have few questions for you. One anonymous attendee asked, have you tried to look at what will happen to the surviving second generation Penium cells without being exposed further to ES7?
So surviving second generation. So if I washed out ES7, but they were affected with ES7, what would happen?
I guess so.
Okay, yeah. So I actually did that. I did a recovery experiment for the paper, and it looks like there is a time that is reached where if the cell is impeded in cell division at a certain point, there's basically no coming back. So even if I put it back in normal media, they will stay stuck together and they'll eventually just die. I've never seen more than two daughter cells stuck together. I know David Domazich there's a couple other drugs that you'll actually have filaments of Penium start to grow, but with ES7, I've only ever seen two. I will say, but before you reach that point, I can get recovery.
All right. Another question, does ES7 also inhibit the deposition of callose at the plasmodesmata after cytokinesis is finished?
It does not affect callose at the plasmodesmata. I don't think we've checked specifically at the wall after cytokinesis. But yeah, it doesn't affect. It's a very interesting drug. It only affects callose deposited at the cell plate. It doesn't affect callose in the vasculature, in wound response. All of those things are normal.
By the way, is it known what is the mode of action of the drug? How it's actually, what is the target?
No, not yet. I think we're close though. So that was the first half of my PhD, is trying to identify the target of ES7. And that's where the collaboration with your lab came into play. I think we're close. We conducted a forward genetic screen in order to identify ES7 tolerant plants. And we have identified several. The one that I identified, I believe, my idea, I didn't get to finish this obviously because I graduated, but the current postdoc is finishing it up. I think it has to do with an ARF-GEF protein, and I think it has to do with the trafficking of the callose synthase itself to the cell plate. But yeah, you'll have to leave it to Georgia's still working on it, so.
Well, I have one more last question from Shunyan Xiao. Do you know which callose synthase in Arabidopsis that make the callose deposited to the cell plates?
We think it's GSL8. That's, yeah, primarily. I don't know if that's been like strictly confirmed or not, but that is like widely, that's kind of the one that everyone's looking at for cytokinesis callose at least.
Okay, and I think the last question that we take is from Laura Klasek. Oh, Laura, hi. So given how evanescent the callose foci in Penium is of a cell plate, can you speculate on other land plant like cytokinesis features that might be found in Penium despite it being unicellular?
Ooh, that's an interesting one. Well, I think the most obvious, what I really wanted to do before I graduated was to look at the cytoskeleton to kind of see what that looks like, because it does have a septum, but it also, if I hypothesize that it has a cell plate, that I think it would have a phragmoplast also to set up that cell plate, but that hasn't been shown. And so I think similarly to Spirogyra, I think that if there's a teeny tiny cell plate, there has to be a teeny tiny phragmoplast that we just haven't seen yet.
All right. Thank you so much. Thank you to our speakers. Katie?
Yeah, I would like to thank you to both of our speakers and also to you and Netson for being such a great moderator. There's been a lot of questions today, so we'll be sure to send those back to the speakers and we can put those on our blog with any follow-up comments that you might want to share. Yes, thank you all for staying, and I hope to see you at our next Plantae webinar. Bye, everyone.
Thank you, Katie.
Thank you.
Thank you, Ning Zheng. Great talk, Destiny.
Thanks.